All right, so we talked about mitosis in our last lecture period. Today we're going to talk about meiosis. And meiosis is a bit different than mitosis and obviously has a much different reason. Mitosis, as you recall, was primarily about what we call somatic cell replication or duplication or forming new daughter cells. These somatic cells are basically cells of your body, so your skin cells and so forth. While meiosis deals with reproductive cells. So we're talking gametes, we're talking sperm cells, we're talking egg cells. We're talking about animals that have sexual reproduction. So that is what meiotic or meiosis cell division is responsible for. It's for making new sperm cells and new egg cells. There's a lot of similarities, but there is some pretty substantial differences. The importance of meiosis is the fact that it helps in, again, having these gametes for sexual reproduction. Sex was an important evolutionary step in making animals or plants or whatever a more diverse, different enough from their parents to take in evolution, for instance. If all the animals or plants were asexual, they're basically clones of their parents. And so they don't evolve as quickly, but with sexual reproduction, you have more diversity in types of offspring, for instance. And those can be selected by natural selection. So again, that's kind of digressing, but we've talked about evolution a little bit earlier. So we're going to talk about meiosis. Meiosis was first observed in Belgium by a cytologist, which is a type of cell biologist, Pierre Joseph van Binden in 1887. So he was one of the first people to really see meiosis, or at least describe it. And again, what meiosis is all about is forming new gametes, where ultimately you have half the complement of chromosomes. Because remember, you get half of your chromosomes from your mother and half from your father. Right now, you're what you call a diploid organism, meaning you have a double set of chromosomes. And so what you're going to end up with is sperm or egg, and we call that haploid, or half the number of chromosomes. So for humans, for instance, you have 23 pairs of chromosomes. So if you count them all up, of course, it'd be 46. And so your sperm or your egg will end up with 23 chromosomes, not pairs. It's not going to be 23 pairs of chromosomes. It'll just be 23 chromosomes. So ultimately, you don't have 46 chromosomes in sperm or egg. You're going to have 23. While your body, as you're sitting here, for the most part, it's going to be 46 chromosomes. Of course, this varies from animal to animal. Then when those gametes come together, we call that fertilization. And so when the sperm fertilizes the egg, the chromosomes join together and it's back to a diploid organism. We call that fertilized egg a zygote. And that's, we'll have 2 copies of each chromosome, one from the mother and one from the father. That may vary from species to species, but that is, for the most part, universally true. And so here is the sperm and egg. Again, they have half the number of chromosomes. In this case, we're looking at 3 chromosomes for the sperm and 3 chromosomes for the egg for this example. And when the sperm fertilizes the egg, it now has 3 pairs of chromosomes, or 6 chromosomes. And so again, that'll be a diploid zygote from 2 haploid sex cells. Now, asexual reproduction wouldn't involve fertilization necessarily. The life cycles of all sexually reproducing organisms follows this basic pattern where you have haploid cells, and then organisms alternate with diploid cells or organisms, because you can actually have a haploid organism. where it kind of looks not like sperm or egg, but actually like in jellyfish, you can have a haploid organism. So some weird things can happen in the plant and animal world, though we're primarily talking about animals in our zoology class. There are three basic types of sexual life cycles. One that you'll be able to obviously identify quickly, and some that you might not. So most animals, for instance, not all animals, but most, you have the sex cells, sperm or the egg, as gametes. And it's usually a very short period. You have meiosis to form those, of course. And then you have the sperm and the egg, and then you have fertilization. You have a diploid zygote. That zygote undergoes cell divisions and so forth. You might have a tadpole ultimately, and then you'll have the adult. And all those cell divisions is mitosis that we've already talked about. But it can change and vary quite a bit. And even in animals, it can vary outside of this. In some cases, for instance, in algae, the haploid cells are actually kind of the adult cells. And it's just a momentary short period of time where you have a zygote. And then it goes into meiosis again. And then you have haploid individuals. So you're a diploid individual, right? But in the case of this algae, it's a haploid individual. So it's kind of odd, isn't it, to think that there's individuals with half the number of chromosomes. And there are some plants and algae, for instance, that have a haploid kind of what would seemingly look like an adult or individual stage. And then they're going to have their gametes and the zygote in a diploid stage. So it's almost like two organisms, even though it's the same one. So essentially, sperm and egg is a living organism. So, and again, this kind of is digressing, but they talk about the debate is when does life begin, and pro-choice and pro-life kind of debates. From a biological perspective, life never really ended. Sperm and egg are both alive. Because remember, the definition of life is homeostasis and replication and so forth. But, and there are some animals that kind of resemble more like this algae stuff. I think bees, for instance, I believe the male drone is a haploid. It's kind of weird. So a lot of weird things after what you're used to as a human and all the animals you're usually thinking about. So obviously this is our sexual life cycle and the animals that we're most familiar with. We have two diploid organisms. They're gametes, meaning they are being formed in their ovaries or testes, and then they get it on, and the sperm and the egg fertilize. This is really reproduction here. It's not all those rated R, rated X movies you see. Reproduction is really when the sperm and the egg get together. That is sexual reproduction. And then you have fertilization and ultimately a zygote. And then, of course, it goes on. So anyway, that's, so let's talk about meiosis. Meiosis consists of multiple cell divisions. So remember, you had mitosis, you had a doubling of DNA, and then it gets divided into two new cells. But we'll double the DNA again in meiosis, but we'll actually divide it 2 times to get the haploid stage. So there's 2 successive divisions, meaning in a row, or over time anyway. The first one will separate the 2 versions of each chromosome. And so that means when you double the chromosomes, they're going to go over as a whole unit. You'll see some pictures, and this will make a lot more sense. And then we have meiosis II, which these chromosomes will then split apart, kind of like what you saw in mitosis. Again, the flow chart will help in a moment. And this will separate the sister chromatids. So basically, again, the end result of these 2 successive divisions is you're going to end up with half the number of chromosomes. So remember, we're going to have something that's going to be a sperm or an egg. Initially, it's diploid, meaning it has two sets of chromosomes. It'll double, so it essentially kind of has 4 chromosomes. Then it'll divide into 2 chromosomes and then divide into 1 chromosome, if you're following the logic. Even when you had mitosis, you had you had 2 in or a pair of chromosomes, but initially you doubled it. So you went from 46 times 2 and then divided it back down to 2 again. But after, does that make sense? Okay, so meiosis I is going to look a lot like what you might have remembered from mitosis. You're going to see chromosomes coiling up again. Because remember, in interphase, you don't see the chromosomes coiling up. And it's actually the longest and most complex stage of meiosis. All these chromosomes are getting coiled up and lined up. And what they call is the homologous chromosomes undergo synapsis. And I'll show you that in a moment too, where they basically pair up along their lengths. What does homo mean? Same. So same kind of chromosome. So let's say you got this chromosome from your mom and this one from your father, and then you doubled it. And then in the cell, they're going to line up next to each other. And then during the first division, they'll be spread apart. And then the next division, they split without putting up the individual fingers. We also have an interesting situation where the chromosomes from the mother and the father can overlap momentarily. And we call this crossing over, so let's say that the purple is the mother and the green is the father chromosomes initially. You can split them, they can be next to each other and they'll split apart and end up with a new chromosome that was neither the mother or the father. It'll be half of each. And I'll show you a picture of that in a second, right here actually. So here you see, let's say again, the purple is the mother chromosome. Now you notice, I want you to get something out of this picture. Notice that this is a chromosome, but it doubled, and that's why you see the other half right here. Because remember, there's a phase where the chromosomes double before all this division takes place. So that's what you're seeing, and they're attached at a centromere. So this is the doubling, and they're still attached together. And so these 2 chromosomes are called sister chromatids. And then here you have the father chromosome, it's doubled. And notice, hey, look, they're close to each other, and this little part breaks off and glues itself back onto this little part. And now we have a chromosome that was neither the mother or the father initially. In fact, it's half, right? So here we got the mother chromatid, and the father's got this little bit in the other half. So what this means is now you have a chromosome that is a mixture of the mother and the father, while this one was just strictly the mother and this one was strictly the father. Now we have one that's primarily the father and a little bit that's primarily the mother. The way this happens in the cells, it's kind of complex. It breaks off, gets reglued by all sorts of enzymes, and things like that. Don't worry so much about that part. Just realize that this crossing over takes place. And why is that significant? It makes those new individuals that come from that egg fertilization have more diversity in their traits. Because remember, these chromosomes represent lots of genes. And this helps to explain why sometimes when we do a Punnett square thing, the individual doesn't come out the way we expected it to. It's more complex than even the Punnett square that you may have learned about in high school because of this kind of crossing over that takes place. So again, the importance of sex is you get individuals that are quite a bit different than their parents. And that allows that species to have a better chance of surviving an environment that's always changing. The Earth is constantly changing. Things like climate change, we may be rapidly increasing it, but that's always been happening. Maybe not as fast as recent years, that's a side topic. But my point is the environment is changing. and the species have to adapt to it. This provides diversity in those individuals that are reborn. And we call this, again, this phenomenon right here, we call crossing over. Does that make sense to everybody? Again, it's, as far as you're concerned, it's just one bit laid over the other bit, and then they kind of re-glued to the other new bit. But obviously, it's a lot of complexity behind that, molecular biochemistry stuff. So this all takes place in prophase 1. That's the one big gist of it. Then we go into metaphase 1. So let's look at, this is what you would see under a microscope, kind of like what you saw when you came to lab. Here you have an interface, a diploid cell. Notice it says it's a germline cell. That means it's a cell that can become a variety of different things, but it's destined to become either sperm or egg. Then the chromosomes begin to coil up, and then we go into prophase. And then notice that this the homologous chromosomes are next to each other. We have the crossing over take place. And then we move into metaphase 1. You remember metaphase in mitosis? Everything was lined up in the middle, remember that? But now the chromosomes are lined up next to each other, on top of each other. Then we have anaphase 1, and the whole chromosome gets pulled to the other side. So that this whole chromosome is going to be pulled over to the other side, and this one will be pulled that side. They're not being split apart yet. The whole chromosome's going. And we call that anaphase 1. Then we move into telophase 1, and it's a lot like telophase that you remember from mitosis. It may be very quick. You might not see, but you may see a momentary nucleus form, and you're going to see cell division beginning, cytokinesis. Then you're going to have cytokinesis and move in here somewhere, and then you're going to move into prophase 2. And then these chromosomes are going to act a lot more like what you saw in mitosis. We're going to move into metaphase 2, and now they're lining up in the middle, just like what we saw in mitosis before. Then they're going to be split into 2. Anaphase 2 is going to look a lot like what we saw in mitosis, but now we have half the number of chromosomes. Because ultimately the chromosomes, here we have 4 sets of chromosomes. Metaphase 1 lines them up. Anaphase 2, we split the 4 into 2. And then when we go into metaphase 2, we split the 2 into 1s, ultimately having 4 cells form from it. So these cells in anaphase 2 now become 4 cells. You can see the little spindles and all that stuff. So meiosis part 2 looks a lot more like mitosis as far as how the chromosomes line up and so forth. And then you have 4 new haploid gametes. So you would have 4 new sperm cells, for instance. Now, it depends on the species. For humans, interestingly enough, you wouldn't have 4 eggs. You would have 1 massive egg and 4 little polar bits of egg that aren't used. And there's kind of support cells. They may be involved with some aspects of the hormone controls, but you end up with 1 big egg and 3 little polar bodies, or whatever they call them. But in some smaller animals that maybe have a slightly different, have a different development, you might have 4 eggs. I'm not really positive about that. But definitely in humans, you end up with 1 big egg. In men, you have 4 sperm. And sperm are a lot smaller than eggs. Again, please take time to read these things, as I usually just verbalize it to you and you all can read. But basically, again, prophase 1, the homologous chromosomes line up. Metaphase 1, or they're made and the crossing over is taking place. Metaphase 1, they're lining up. Anaphase, they pull apart. And then telophase, we get some cell membranes, maybe a nuclear membrane. And then we go into, we're going to go back into prophase 2. So here's the picture of the actual cells, and then this is the description down here. Here you got your centrioles. I hope you remember that. And you can see some spindle fibers coming off of it. You can see the homologous chromosomes beginning to form. That's how you know you're going into prophase 1. You're seeing the crossing over here, right? And then you see the, in metaphase 1, the homologous chromosomes lining up. Anaphase 1, the chromosomes are pulled apart. Telophase 1, you see the chromosomes kind of relax a little bit. You see the maybe the beginning of the nuclear membrane and cytokinesis taking place. Or cytokinesis is dividing the cell. Then we go back into, we're ultimately going to go back into prophase 2. Here you can see in the picture the cell becoming 2 new cells, cytokinesis, interkinesis, the nucleuses, all that stuff is kind of forming. And then in meiosis 2, we're not going to have, now remember an interphase, when we're doing mitosis, interphase, we doubled the number of chromosomes in mitosis. Meiosis 1 or prophase 1 or interphase 1, we double the chromosomes. Interphase 2, we don't double the chromosomes. We just leave them alone. Okay, does that make sense, everybody? So again, once we start dividing after that initial doubling, that you don't see that happen anymore. And of course, the crossing over takes place. So again, prophase 2 is a bit more, in appearance-wise, looks more like mitosis. Prophase 2 is much simpler than prophase 1. Remember prophase 1, you got the sister chromatids crossing, all sorts of stuff. It's much shorter in meiosis 2. The spindle fibers bind to both sides of the centromeres, line them up in the middle, and then again, anaphases pulls them apart. So I'm just kind of repeating the same things over and over in hopes that maybe it sinks in a little bit if it's a little confusing to you. Obviously, the nuclear membrane is going to reform and so forth. So let's take another look at these pictures. Okay, so we've already gone through prophase 1, metaphase 1, anaphase 1, telophase 1, interkinesis. Now we're into prophase 2. We've had this brief interphase. We start to see the chromosomes forming again. We call that prophase 2. This time, the chromosomes line up in the middle. We call that metaphase 2. And then you see the cell membranes already here between these 2 cells. Then we have anaphase 2, and then a cell membrane forms between them. And now you have those 4 cells. In the case of men, again, it'll be sperm cells. There might be some more development stuff, kind of things happening to make the sperm cell. This is the meiosis that took place. Again, you're going to have half the number of chromosomes, so they're haploid, and you may have some unique chromosomes that were never originally in the mother or the father, because you've now through meiosis have modified some of the chromosomes to be half and half. So you don't just get, so you know how we talk about you get half the chromosomes from your mother and half from your father? That's not exactly true, right? Because in some cases, you may have a few extra bits, depending on how this meiosis took place. So here's some more pictures. Well, you still will, but my point is that you get some unique chromosomes that were neither the mother or the father. They're now a mixture of the two. Does that make sense from the colors you're seeing? Again, I show a lot of pictures for you to look at it over and over again and try to get an idea of what's going on. So it's just reinforcement. Take time to read it, look at it, watch the video again, but it helps to reinforce it, especially since I don't necessarily give you a textbook, but you can find this information just about any decent textbook. The stuff's been around for a while. But again, the big thing about meiosis and sex is it really increases genetic diversity. It's shuffling cards, for starters. Imagine if you had 46 cards and you shuffle them. You're gonna get a different kind of deck, right? From your sister or brother. Then you have this crossing over. The independent assortment is like shuffling cards. Then you get this crossing over. And then the sperm fertilized from two different individuals. And now you have an individual that, is different in many ways from their parents. On top of that, again, the sperm and egg are running around fertilizing. There's some randomness going on there as well. So this is showing you some of the independent assortment, like you have this paternal gametes and your maternal gametes, and they come together as, in a diploid individual. And so again, it's a whole different assortment of chromosomes that may not have, that didn't exist before in the parents or the mother. So the point is, from this slide is this: there's such a multitude of potential gametes that would come out of these individuals. So this is your mother, this was your father, this was your mother, this is you, and then this is your gametes, whether they be sperm or egg. You'll have a huge different assortment of potential gametes than what you were as an individual. Does that make sense? So look at this one. In this case, this sperm from the father is represented more so in the sperm. Does that make sense? See, they're all purple. This time we call them purple than that. This time all the green. So you can have sperm or eggs that represent more your mother or your father because of this independent assortment. Does that make sense? So it is true that some, again, it's so many genes, but some features you may say, oh, I really got that feature from my mom, from my dad. And then what you pass on to your kids are going to be different too. So again, it's just a huge, 2 to the 23rd power possibilities. 2 pairs, right? 8,388,608 possible kinds of gametes. And then again, you got this DNA crossing over. So you have new chromosomes that never existed before because of now you have a male and female chromosome, essentially, in quotes, that's what I'm calling it. One from your paternal side and one from your maternal side. So again, when you think about these different possibilities, and then you hook up with your partner, you're just talking trillions of possibilities. So think about how lucky you are to be who you are. One little difference in your sperm and you wouldn't have existed, right? You won the lottery. Really. Not even just talking about genetically what you are, but who you are, where you are. Let's see. Again, I think I've hammered this enough, but genetic diversity helps in species survival and adapting to different environments. This is a big part of why evolution exists, because there is variation. I think you got some, when I was going through this, I was kind of giving some comparisons to meiosis and mitosis. And this is some take-home messages. For the most part, you're not going to see crossing over in mitosis. You don't have you're not going to have chromosomes lining up. So you're not going to have the synapses going on. You're also not going to see a reduction in division. I mean, you're going to double the number of chromosomes as a diploid somatic cell in your skin temporarily. You go up to 46 and then come back down to 23. But ultimately, you're going to have the same number of chromosomes as that you started with. So you start out with 23 pairs. You're going to end up with 2 cells with 23 pairs. So you don't have this real reduction going on like you do in meiosis, where you ultimately have half the number of chromosomes. So those are the big differences. So again, I feel like I've hammered this and explained this enough to you. Here's our diploid and different phases. So again, I think you got the gist of this, but look at this slide here. This may be worthwhile. Here we have our meiosis and our mitosis. Here so far, everything's the same. We doubled the number of chromosomes. Then we go into meiosis I and prophase I, and you got the crossing over, and you got your chromosomes crossing, being pulled apart in metaphase I and anaphase I. And then you got telophase I, and then you go do that again. Well, over here, mitosis, all that is skipped, and we just essentially go into metaphase and line up the chromosome. So does this help a little bit? I'm hoping you got an idea for me, because I'm kind of just ultimately saying the same thing over to you. But in the end, mitosis, you end up with 2 daughter cells with the same number of chromosomes as the parent cell. In meiosis, you end up with 4 haploid cells with half the number of chromosomes as the parent. All right, so that wraps up meiosis, enough to get you make you dangerous, and an intro to zoology class. And I appreciate you listening, and I will post this for you. Thank you so much. Let's watch